All right. I mean, I want to tell you a little bit about how I got into this. I was interested in fish from a very young age. That's me there with my mom, who was the real fisherman of the family, and also interested in nature. As I went through college, I used to have some weird pets, like an iguana, and I took this interest in organisms and biodiversity and began to actually study it out in the wild and started exploring areas of South America like uh, Peru and like Guyana in 1998. Well, we realized we needed to get more and more into deeper and darker areas to really figure out things. And so this culminated in this trip to Guyana in 1998 to Kaiteur Falls. But we also explore biodiversity in other ways as well. For instance, we've been exploring cave biodiversity. In particular, there's cave fishes in northern Alabama and Tennessee and Kentucky and a few other places, and we've been going into those caves and actually finding out what's actually living inside of there. And you can see me in one of those caves. We've also worked across the world. This is Democratic Republic Congo, an area known as Kisangani. This was made famous by the Heart of Darkness novel. And here's Guinea. We're showing you one of the things that we use. So actually, this is actually using an electrofishing rig where we can put electricity in the water and then shock the fishes to catch them. And this is a gill net, which we often use for things that are harder to get in deeper waters. We went to Papua New Guinea. One of my students was studying marine catfishes, and there's a large freshwater radiation of marine catfishes in Papua New Guinea. And what I do is I put these things, put anatomy together and put genetics together to figure out how things are related to one another. And we express these in trees like this that are called phylogenetic trees. And this is kind of expressing that this orange group here has this very highly modified air-breathing stomach in this particular group. And this group that I study is actually made up of the Suckermouth armored catfishes. And what is one of my primary responsibilities is actually going out, discovering, and describing new biodiversity. And so far, I've described about 60 different species, almost all within this loricariid group, except for one little minnow here. And that's kind of telling that minnow is actually from right around where I live in Auburn, Alabama. So there's even new species being discovered here within the United States. But most of these are from South America. And to put it in perspective, I've described about 6% of the total diversity of this particular family. So that's a pretty large amount within the last 20, 25 years or so. Now, most people may not know that they have a familiarity with the Guyana Shield region. But if you ever read The Lost World or saw the various movies that have been done of this, what you know is you know a little bit about that particular area. Doyle was inspired by a formation called Mount Roraima, which is in this particular region, and he imagined dinosaurs on top of there because this was such a remote area, and you know if anything was going to have dinosaurs on it on the planet, it was the remoteness of the Guyana Shield. For those younger, maybe you know it from Up. So Up was based upon tapuis that are found in on the Venezuelan side, but similarly occur on the Guyana side as well. But this is what it really looks like. So this is Kaiteur Falls. It's one of the largest waterfalls in the world. It's the largest single drop waterfall. It's about 750 feet tall. And our first trip there was in 1998. And this was actually with National Geographic. It made it in the magazine in 2002. And they said Jonathan Armbruster, a jungle river vendor, and slithered on his ample belly along the stream bottom, a swim mask on his face. So I'm kind of infamous for that. And they also talk about me kissing a fish in this. That may or may not be true. Anyway, what we did with that was we actually went back to an area that was first explored in 1908, and nobody had been back there since. And we looked at distribution patterns. So the Kaiteur Falls actually doesn't change the amount of species that are in the stream that much. What did change it was the first falls on that river, which is called the Kaiteur River. So what I'm going to talk to you today about is this area called the Guyana Shield. I'll then focus in on our work in the Rupununi portal. That's more ecological type work. Excuse me, my throat's a little sore today. Then I'll talk about this ancient river system called the Pearl of Berbice. And then I'll kind of try to tie it all together with some high shield biodiversity and some of the trips that we took to some of these areas. So, Guyana Shield. This is roughly in red, the Guyana Shield here. And we can break this up into quadrants. And we've done a lot of work over here in Guyana and Venezuela. We've had colleagues that have worked a lot over here in Suriname and French Guiana, as well as the Brazilian state of Amapá. But all of this area down here along the southern portion of the Brazilian Shield, most of that has not been explored at all. So what's interesting about this area is that it's an evolutionary museum in that it preserves ancient lineages. So this guy here, Lithogenes, it might not be a dinosaur, but it is sister to everything else in its family. So in other words, it is sort of a prototypical member of its family, and it's isolated in a couple places on the Guyana Shield. There's also some highly adapted things here. So this guy is very highly adapted to fast water, and it's very similar to some things that you find in the Andes. And then we also see some things that actually occur throughout the Guyana Shield, and then also into the Brazilian Shield, which is located south of the Amazon. So here's the Amazon River here. This is the Negro River going this way. This is the Orinoco, and this is the Essequibo. All those rivers are going to be important as we go through here. So what's interesting about this area is that the Amazon is like a parasite. It's stealing rivers from other river basins. And there is an area in Venezuela here where 75% of the upper Orinoco River is actually diverted through the Casiquiare River into the Amazon. Eventually, that whole upper Orinoco will be stolen. Similarly, in our area over here, there's the Rupununi Portal, and that area seasonably floods and allows connection between the Sacutu Bronco Negro system that goes down in the Amazon and the Essequibo system. So I'm going to talk about this more in detail, but through most of the Cenozoic, so for most of the past 65 million years, this is what the river systems look like. There was this proto-Berbice river system. This has been built together by geological evidence, and we're providing the biological evidence that this river actually existed. Anyway, let's talk about the Rupununi Portal and some of our ecological work down there. So the Rupununi Portal is in an area known as the Takutu Graben. A graben is an area between uplifts of continental material. And throughout most of the Mesozoic, so when the dinosaurs were around, this was actually a lake. It was called the Paleo Lake Mericanta. And that over time what happened is that the Pacaraima Mountains up here in the north and the Kanuku Mountains down here in the south eroded, and all of that material filled in the gap here, filled in that lake, and that changed this area from a lake to a river system right around 65 million years ago. Today, this central area is known as Lake Amuku, and this is a seasonably flooded lake that is dry during the dry season and is totally wet during the wet season, and we shall see. So to give you an idea what this looks like, this is savanna region. So normally when you think about South America, you think about rainforest, but some of the best areas for fishes are actually in the savanna regions. What you're seeing here is the northern end of the Rupununi savanna, and up above here are the lower parts of the Pacaraima Mountains. This is on the other side. These are the Kanuku Mountains. So we're just south of the Kanuku Mountains. And this is an area called Sand Creek that we'll come back to. But you can see this rock here is really, really old. And it's really hard. There's not a lot of habitat in some places. And it's a difficult place to work. And this is Lake Amuku. It doesn't look like much of a lake right here. This is the dry season. We like to go during the dry season because the fishes are concentrated into areas and it's a lot easier to catch them. Because this is what it looks like during the wet season. You can see the Pacaraima Mountains out here in the distance. This whole area floods. And you can imagine fishes that don't mind lake-like conditions can move across here. In fact, this area is a major nursery ground for a lot of those fishes and an important area for this connection between the Amazon and Essequibo river systems. But it's also almost impossible to work at this time of year. So, what kind of led us into this is we want to know what was out there. That's sort of the key of biodiversity, is knowing what's out there. And we don't have great information on fishes. And we wanted to see if there were differences between the Amazon side, or the Takutu River, and the Essequibo side, the Rupununi River. And this is potentially to identify areas of conservation concern. So we looked at this through various different types of statistical tests, And we've done at least 4 major expeditions down there, resulting in 433 species, greater than 52,000 specimens, and 132 localities. And sometimes you have to watch out for the local predatory fauna. This is a black caiman. Black caimans get a little bit larger than American alligators and are more aggressive. Anyway, you can look at this in a Venn diagram form here. So this area in light green here, this is the species that are shared between the two basins. We would expect that because they were formerly connected and they're at least partially connected now. But what has happened in the Takatu is that it's been enriched by Amazonian fauna. There are unique things that occur only in the Amazon, and they are in the Takatu but haven't made across. Some have made it into the Rupununi, and that's within that shared pile. The Rupununi, however, retains more of the proto-Berbice specimen, so species, things left over from that old ancient river system. So kind of curious about what has crossed here. So you may have heard the legend of a kangaroo. Kangaroos are catfishes that swim into fish gills, and they have a little tooth plate up there, and they grab onto one of the large blood vessels in the gill chamber and drink all the blood out of it. Anyway, the large ones, which probably rode on larger catfishes across the Rupununi portal, are found in the Rupununi. But the smaller ones, which fill up and drop off a lot faster, have not been able to move. So there's a lot of ecology tied up in what's able to move. And so this is a way of looking at what's called a species accumulation curve. You want to know if you've gotten everything that's there. And what this shows you is that since we've hit this line on all the different types of metrics you can look at, they're way out here, basically no more sampling is going to add a significant number of species that we don't already have. So we pretty much have sampled the complete fauna. We have a couple little things out there, but for the most part, we've gotten everything. So this is actually taking a look at how communities are similar to one another. And so there is sort of a difference here between the Rupununi River in the red triangles and the Takatu River in the blue triangles. This area where there's kind of overlapping is that area that is Lake Amuku, where the species are expected to be more familiar. But we noticed a few other areas that weren't in that particular region. And so what we did is what's called a clustering analysis. And this just shows you the similarity between different areas. And what we found is that there were these clusters of Takatu and Rupununi specimens that were not, or localities that were not in that Rupununi portal region. So what we found out was that from the locals down there is that this area also seasonably floods. And this is a second place where fishes can move across between the Essequibo and the Takatu system. So anyway, what we're looking at here is this is where that Sand Creek portal is. You can see how close the river systems get to one another. So it's very easy to move across. The red dots and white triangles or white squares are all of the areas that we have collected. The white squares are where we've gotten 75 or more species. There's an area in Caranambo here, the most speciose place I've ever been to, somewhere around, I think we got 98 species in that area. But anyway, we put this all together, and we were proposing to the Guyana government that this area become a Rupununi Savanna Preserve. And there's some reasons for this. Brazil wanted a port to the Caribbean, so they built a large road that goes right through this area. Oil was also discovered in this area, but it turns out it's not too economical to actually extract it out of here, so there's not much going on there. There is a lot of farming that goes on here, particularly cattle ranching, and this area is very important to the maintenance of fish stocks of the larger catfishes and other fishes that people eat, paimas, for instance. And so we're suggesting that they preserve this area. And my former student, Leslie D'Souza here, has actually met with the president of Guyana to discuss things like this. All right, let's talk about that proto-Berbice. So what we're looking at here is northern South America, and these are all the major rivers here. And once upon a time, even areas of the upper Orinoco here and here, this is the Ventuari River that's going to become important. Most of these major rivers went out through what's now the Berbice River. And we know this in part because the Berbice has a delta that is not of a size of that river. So the delta is tremendous in size, but the river is tiny. And so this suggests that there was a much larger river that went through there. And so the geologists have put together a scenario and then they kind of rely on us to actually go in and figure out if how much similarity is present within these various different river basins. Excuse me. So through most of the Cenozoic, so for most of the last 65 million years, what we have is that upper Orinoco tributaries here and here all flowed into tributaries that are now going into the Amazon and eventually joined the upper Essequibo River system, the Corentine River system, and eventually the Berbice River system that went out here. Whereas things like the Cuyuni, the Mazaruni, the Pataro, and the lower Essequibo all had a separate mouth. So somewhere along the line, these guys here flipped into the Orinoco River system. I'll talk more about that a little bit later. But about 5 million years ago, the Bronco steals the Araraquara River here and the Cotinga River. About 2 million years ago, the Amazon steals the Takutu and the Ireng River system. And somewhere around then, too, we switched into this modern distribution of those rivers. But this is an ongoing process, and the Amazon is actually eating its way through, and that's what the Rupununi portal is kind of telling us, is that eventually the Amazon will steal the Rupununi and maybe even the whole upper Essequibo River system. So these are important for understanding how fishes are distributed across this area and how we can actually figure out how things are related to one another based upon the geography. So we come up with scenarios about how fishes could potentially move across these various different areas and look at it with the relationships of the fishes as well as distributions. And you can come up with fancy diagrams like this that tell you about when we expect different areas to have split, so we can look at the genetics of organisms and actually come up with a tree that seems to match this proposed, it's called a species area relationship. Excuse me. So one of the first things, the thing that I couldn't wrap my mind around was how did these streams that are now part of the upper Orinoco, how did they switch from the Proto-Berbice into the upper Orinoco? And this is actually Venezuela here with one of the major waterfalls that's present there. And we see some things like this. So this is the genus Chaetostoma. Almost all the species of Chaetostoma are found in the Andes, and there's a couple down here in the lower Amazon as well. But how do we explain this distribution? These are called rubber-nosed plecos, by the way, in the Petraea. Also some cichlids. There was this weird distribution, particularly in the Cowra River, where the upper Cowra is a different group of species than the lower Cowra. How do you explain that? Well, it turns out there's actually these transcurrent or major faults that are present in this particular area. And what this did was lift the Pachyramas haven't been quiescent. They've actually been moving around a little bit in the last years. Even though these mountains are on the order of 4 billion years old, there has been tilting that switched those areas up. All right, somebody did ask, is there a lot of competition between these fish? And the answer is yes. This is how we explain why there are so many fishes here, is that in South America, fishes actually can divvy up the resources much more than what you can do in, say, North America. So, for instance, algae scraping, which is one of the things that my group of fishes do, they up here we have like one species that does that, but down there we have things that are specialized on scraping on rocks, scraping on trees, scraping on leaves, scraping gravel, scraping in fast flow, slow flow, all of those different areas. All right, so this was a major area that kind of just flipped everything into the Orinoco. So we lay these fault lines on that map. What we see is that that's pretty much what's explaining why those fishes are different in that Cowra River. That break is right at that point. And similarly, how do we have Chaetostoma on both sides of these mountains? We had it because these tributaries here flipped over, and then somewhere around 5 million years ago, these guys split and became their own separate things. All right, we wanted to take a look at this with some other groups of fishes as well. So we actually went to the Ireng River and we caught one of these guys from that area, but it turns out it's actually not related to these. It's related to things over there, just making life even more confusing. All right, what I want to spend the bulk of this talk talking about, though, is some of the data that we actually got on this high shield biodiversity and talk about the trips that we took to actually find these things. So what I'm going to talk about are several groups of fishes. So we're going to talk about these Suckermouth armored catfishes called Lithoxini. They're really flat, small things that live under rocks. We're going to talk about this other group called the Corymbophanini, and these are larger fishes. We're going to talk about the pencil catfishes. These are related to those kangaroos I talked about. The legend of the kangaroo, by the way, is that they've been known to swim up human urethras instead of fish gills. And there is pretty good evidence that that has actually happened. It's not great for the fish either. And then we'll also talk about these guys, which are the South American darters. So we've taken several expeditions to the High Shield, which is some of the most remote areas on the planet. And, you know, seeing it, you can imagine there are dinosaurs or giant flightless birds up there. Not, but there are some really cool organisms up in these particular areas. So I'm going to talk to you about this trip here. This is the Currabong River. It joins the Putara River here and goes into the Essequibo River. And we were sent in by the Guyanese government because of this fish. So the Guyanese government wanted to put a hydroelectric plant up there. And one of my former students went up there and discovered this particular species of South American darter that was found only up there. And so they wanted us to actually see if there was a greater range of these fishes in that area. So we came in and we actually flew into Kaiteur Falls, and the water looked like this. So we as ichthyologists want to go to these areas when there's low flow, because getting out in water like this, not that we would do it right at the edge of the waterfall, is dangerous. You can die from this. And to just give you an idea of how much water is going over here, I got a little video of this. Maybe it'll work. Oh, wait a minute. Put on the. I'm not sure how to turn that on, but it is really loud. But there we go. That's what you experience. And as a fish person, you look at this and you're like, oh no, there's no way we're going to find any place to actually work in this particular area. So there you're out looking down the Putara River gorge there. And this is a national park, but it's not like North America. There are no handrails anywhere. Okay, let's turn off that sound. Oops, maybe we're not. All right, so we actually were able to get on, hitch a ride from the river. And we came into the Currabong, and it looked like this. Again, this is stuff that kills people. This is really, really dangerous places to work. So just kind of looking at, this is our helicopter pad I'm taking a picture from, and you can see how the helicopter approaches in there. Not exactly an easy place to get into. This is what the Currabong River looked like down there. And again, we can kind of look at this with sound too. Maybe not on this one. All right. Unlike Kaiteur Falls, you can't get a really good view of Amila Falls, but this is the edge before it drops off. It's not quite as impressive as Kaiteur Falls, but it's still a pretty big waterfall. Now, we did find this area that looked like it was shallow. There's these big plants in here. We call them troll grass because it kind of looks like those troll dolls, you know, with the hair. And normally troll grass is about 6 inches or so, but for some reason here it's gigantic. So I'm actually standing in a precisely John-sized hole here and holding up one of these things. The stalks on them were about 3 feet long, so moving around here was really, really difficult. Next step. Not moving on. Oh, there we go. And the other plants in here were this plant called pacu weed. And this was in a lot of the flowing areas. This kind of what it looks like underneath the water. It has little bladders that helps them float, and their stems are kind of air-filled as well. And when they dry out on the rocks is when flowers come out of the stalks, and they're not very pretty flowers. What they are is they're fly pollinated, so they don't smell very good, but the flies come in here and pollinate these in the brief periods when these fronds come out of the water. So it was a bizarre area, full of holes, lots of difficult areas to work. But as time went on, it became easier to work because the water here flows off very quickly. So that by the time we actually left the upper curve rung to go down to the lower curve rung, this is what the river looked like. It had almost completely dried out by this particular time. But the water doesn't stop; it's actually flowing subterraneously at this particular point. All right, so from there we had to actually hike down the mountain to the other side. Somebody asked if there are any poisonous fish in the area. Poisonous, no. Venomous, yes. A lot of the catfishes have a venom in their spines, and the difference between a venom and poison is a venom is injected into you by the animal. But the only ones are the catfishes, and you'll see a couple of those hopefully in a little bit. So we were up here, and we had to get down here, and that's not an easy area. We didn't obviously traverse that. There are areas and cracks that you can kind of wind your way down to the lower portion of the curve on. As for some of the things we got there, this is a species of South American darter that was described by that guy Eigenmann, who had gone to this area in 1908. Headstanders, these guys are quite beautiful. These are both of these fishes are about 2-3 inches long. This is a species of bristlenose pleco. We just described the species last week, so it just got its name Kaiteur last week. This is one of the catfishes that do sting. It's one of the 3 barbed catfishes, and they have stingers on the pectoral spines as well as the dorsal spines here, and some of them can hurt quite a bit. Corridor's catfish, actually some of these guys sting too. I don't think this one does, but this guy's only about 1 inch long or so, and people buy these for their aquarium all the time. Probably the most beautiful fish I think I've ever seen is this guy. He's about 2 inches long. Typhlichthys Eos, and Eos means dawn, so we call this the dawn tetra. It's just a lovely thing that only occurs in these little pools in the rainforest. Lebiacina, it's a shame to call these pencilfish. I mean, they're part of the group that includes the pencilfish. These guys get pretty big. They get about 1 foot long or so, and almost all of them that we find are new species. They have very small ranges, and this is certainly a species that is new to science. What's kind of interesting is I was kind of trying to find common names of these things and looking it up on Google, and my pictures were coming up on as the pictures from Wikipedia. So somebody's mined my photos, and you can see a lot of these things on Wikipedia now. This is a really cool one. It's called the cat's-eyed catfish, and you can see that that slit pupil there. It's a really unique fish. This is probably the best picture I've ever taken one of those, if I do say so myself. A lot of the tetras down here kind of have this orangish brownish tint, and the reason why is that this water here is black water. It has a very, very black tint to it. Kind of looks like coffee, and this particular color pattern kind of helps hide them within that coffee-colored area. Okay, somebody asked how many different fish live in this area. It depends on where you are. So on the top of the mountains, there's only about 20-30 species. You get down into the lowlands of the Rupununi, we're talking hundreds. 430 species, I think, is what we found. Here's Cope's tetra showing about the same thing. This is a weird little dwarf cichlid that's only found up there in that particular region. Oops, the name's kind of slid across here. This is a miniature catfish called Cichlids. They sometimes sell them in the pet trade, but this particular species is incredibly rare. We've only collected it a couple times in Guyana, called Cichlids collinsae. These are another type of South American darter. These guys are more midwater, and it's a weird situation where both the males and the females are pretty, but they're pretty in different ways. So this is the male. He's got a red anal fin here, whereas the females have this checkerboard pattern instead. And this is us after climbing back up the mountain into one of these high montane streams. And this is the type of habitat that we have to work in. This is among the hardest habitats to work in. We can't use electrofishing here because there's not enough ions in the water to electrofish, so it's all hard work getting things out of here. Now, the reason for doing this, though, is that this area is being impacted. It's not being impacted by lumber. It's being impacted by the mining industry, particularly gold and diamond mining. And the lower Currabong is among the most impacted areas for gold mining in the world. And this is just the start of that activity that we got down to. And just to show you what a gold mining area looks like, this is it from the air, and this is a relatively small mine. This area that they've turned up here, this is area that will be toxic for generations, probably for hundreds, perhaps thousands of years. Nothing will be able to grow there because of the toxic metals that they have stirred up in this particular area. So you can see that rainforest is almost perfectly intact, but these areas have been very highly mined. And what the long story of this is that our data show that probably shouldn't build a hydroelectric plant in this particular area. You would cause you would threaten the continuance of several species. And the problem is that they built a road to the base of the Mila Falls, and that has allowed miners in. And so this is the first miner that actually comes in here. I have a few more questions here. How many species of fish have you discovered? That was 60. Is it possible that the waterfall harms fish when they fall? Yes. So if a fish goes out of the waterfall, it's probably toast. We have seen some interconnectedness genetically between the lower portion of the falls and the upper portion of the falls, but it's not very recent. But what you realize is that the falls are what you see now. When there's high water, the water goes around the falls, and there are more gentle slopes in other areas. Just to show you the remnants of a mining camp. This is one in Suriname that we actually walked down into, and this is among the creepiest sights I've ever seen in my life. They just left things behind, and somebody created this shrine to Santa Claus, some monster thing, and a teddy bear. So these are some creepy areas that you go to, and they really do destroy the habitat. Anyway, when I eventually flew out of the Currabong system, this is what it looked like. It was down to a trickle inside of that stream. And this rock here is very, very hard. So water goes in very quickly. The water goes up very quickly, but it also goes down very quickly. So we were able to find places to work even when it was high. And then when it got low, it became a lot easier. All right, the next trip is to the Ireng River. And this was to Orunduk Falls, which is a step waterfall. This is the lower step. And this is our crew. The rock is this red jasper. Jasper is a really, really dense rock. It's actually a semi-precious mineral. People make art out of it. Okay, we're loading the second part. And this is the upper part of Orunduk here. And then we boated from there up to the Amerindian village of Kaiparopai. Amerindian is the term that people use there collectively for the groups of native peoples. This particular area is run by the Patamona Amerindians, and they are really fantastic people, really know how to work and live in the jungle. This is the falls of the Sarama here. Beautiful, beautiful place. Anyway, sometimes it's best to see these things from the air, and we had a drone with us, so you can see what Orunduk looks like. It's, again, this might be a little choppy in the video here, but you'll kind of get the picture of it. It's a savanna. There's very, very few trees in this area. This area is in a rain island, and we had to boat up about 6 hours before we got into rainforest. All right, somebody asked how many catfish species are there. That's a good question. 10 or so, up in the highlands. In the lowlands, 100. All right, so this looks fairly shallow, like the water type of water we want to work. But the problem is it's all flat rock like this without any habitat. But the only place we could find up in this region to actually work and find fish was in this area right here, because there's boulder and cobbles that can provide habitat for those fishes. There's a little guest house over here where we stayed, but the shower facility was right there. Oh, I'm sorry, I don't have my pointer on. The shower facility was right there. That's better than most places I've been. All right, I don't think he's going to show up. Okay, maybe the last video will show up for that. Anyway, what did we catch up there? We got a lot of tetras, so these are sort of like the minnows of the area. These are your common morphologies. Bricon americus, this is one of your generic tetras that you find all throughout South America. You get some of these traheras or wolf fishes. This is a smaller one. They only get about 6 inches long or so. They're weird in that they actually can breathe air. They do it with their swim bladder. Related to that is a larger thing. This is Hoplariothrynus, but doesn't seem to be showing up for us. Knife fishes, these are weird electric fishes. They're weakly electric fishes. They produce a current that can actually be used for navigation. We did get some electric eels in some areas too, and I've been shocked by a few of those. Cichlids, call them sunfishes down there. We call this a pike cichlid. They can be beautiful predatory fishes that get about 6 inches long or so. This is the keyhole cichlids. They're common in the pet trade, although this one never makes it there. This is probably a whole species complex of a bunch of different undescribed species. Some little top-dwelling fishes like Pyrrolina here. And then these kind of algae scraping tetra-like fishes as well. They have a little modified jaw to scrape rocks, and I just accidentally put my camera down and managed to capture one of those swimming through the water. Somebody asked what inspired me to study fishes. There was a lot of things. I just was really interested in fish when I was going fishing with my mom as a little kid. Then in high school, I started keeping fish and then worked in a pet store. I actually started off in engineering and then decided I wanted to be outside and became an ichthyologist instead. Excuse me. This is a type of catfish. This group is really unknown. I actually have a student from Peru coming to visit me this week to study our specimens of this particular group. This is another species we just described last week called Ancestry saudages. This is another type of bristlenose plecostomus. And then swamp eels. So swamp eels have been in the news because a few of these species have been introduced into North America. This particular one is not. This, again, is a species complex of a whole bunch of undescribed things. So let's get into some of the actual science of this all. So we've looked at this group, these really 2 inches or so small flattened fishes called the lithoxini. And so small flattened fishes called the lithoxini. And so we've been able to gather specimens from a lot of different areas throughout northern South America, and then recently discovered this group is actually also down here along the southern part of the Guyana Shield. And so what we decided, this is a tree of relationships based on genetics. And what we found was this one species here that we had described in Lithoxis actually was sister to everybody else and required a new genus name, and we called it Avalithoxis. And this is an interesting thing. It is, again, one of the most remote areas that you can get to. We only got a few specimens of it, and there it is. Now, usually morphological trees actually have very different relationships because there's a lot of convergence in morphology. So two different things gaining the same character. But in this particular case, came out exactly the same. So this was actually sister to everybody else. Somebody asked me about what the function of the bristles are. So in those bristlenose plecostomus, they have this big set of snout tentacles. We're not quite sure. We've hypothesized that they're larval mimics because the males actually take care of the eggs and then the larvae until they absorb their yolk sac, and they look like little larvae sticking on top of the head. One study, and unpublished, actually found that there are these little cells on the inside of the tentacles that produce a glycoprotein, a type of mucus. And the same glycoprotein was found in the bellies of the babies. So there's a potential, and again, we don't know if this is true or not, that they actually feed their young. Anyway, we can actually throw our tree of relationships on the distribution, and this is kind of hard to read, but one thing we see here is that one of the reasons we went to the upper eye ring was to catch this Lithoxis bovalii, and things were called bovalii from several different areas across this region. And what we found was that the specimens from the Ireng were sister to the specimens from the Quarantine, and not to ones that are actually closer to it. But if we remember, this and this were all part of the Proto-Berbice system, but the Petawa River system here was not. And we're kind of seeing that relationship still within this particular tree. All right, somebody asked why some of the species may be adapted to have brighter colors. The reason why is that colors are generally signaling between from males to females or between males for competition reasons. So for the most part, just like in birds, the colorful individuals are the males. Similarly, if we look at this here, our Exastilithos that we got in the Ventuari River above a waterfalls here was actually sister to something way over on the other side. Now, nobody's been anywhere between here, but this again supports our protoburbese hypothesis because not too long ago, these two areas were actually connected. Then there was this fish we turned up in the orang, and I didn't know what it was. I'm supposed to be the expert on this, and I didn't know what this particular species was. But there are a few cues. It doesn't have an adipose fin, which is a little fin on the back here that catfishes usually have. Instead, it's been replaced by a ridge of plates. On the cheek here, these fish actually have teeth that stick out of the cheek. Actually, the whole body is covered in those teeth. Excuse me. They just get a little bigger in this particular area. And then its eye was weird by being normal. So most of these fishes actually have this flap of the iris, but these guys have a nice, perfectly round iris. Now, my first guess was Nebulonichthys, and somehow this got moved over, but this is a group that is found in this particular area. We would have expected to find it. What's weird about these guys is those teeth get really, really big on the snouts of the males. And again, we're not sure what they do with them, but they probably do fight with them, or at least maintain territories. But the other choice was this guy, which is Corymbophanes. This was a species that we had gone to get in the Petawa River. This was the species I kissed the lips of because nobody had seen it in 90 years because nobody had been back there. And it similarly had no adipose fin and had a ridge of plates there. It didn't have cheek odontodes here at all, though. But its eye was, again, weird by being normal. Somebody asked, do you think there are more undiscovered species of fish in the areas you have studied? Absolutely. Most, I understand one group of fish. I don't understand them all, and we're still describing new stuff out of what we've described, and we send these specimens all around the world to actually identify. Now, these guys, like the Nebulonichthys, also get these really big teeth. We only have ever found one adult male of these guys, and it was found dead, floating. And it has these tremendous teeth sticking out of the snout here. And these are just the same material of teeth. They're enamel, they're dentine, they're homologous, or the same thing as teeth. So, unfortunately, these two specimens we got out of the orang were both females. And as we picked them up, eggs were coming out of them. And so there were clearly males in there as well, and we just did not get them. And it's kind of a shame because they probably look like this. But anyway, this group, Corymbophanes, I thought was related to everybody else and was like a really basal member, whereas Nebulonichthys was up here with a whole bunch of other things that have these evertable large cheek odontodes. But when we first did the molecular phylogeny, we actually found Corymbophanes sister to this thing. Not only does it have cheek odontodes, it has like these huge claws sticking out of the side of its head. This is for protection. You can kind of wedge yourself into a hole with these, and then you pick them up and they jam those things right in your hand. They kind of hurt. But here's Nebulonichthys here, and it's actually right in the same region of that tree, so it's probably closely related. So we've actually gone in and gotten greater detail of the phylogeny here. What we found out was that our species that was in the upper curve rung was different from the species that Eigenmann described here, Andersoni, as well as the species we described from the Loricariidae. So we're describing it as Corymbophanes ameliae. Mila Falls is named after an Amerindian girl who died in that region called Amelia, and so we're going to name it after her. Similarly, Kai is named after an Amerindian luncheon. Sister to that, it says Irenichthys, but we're actually going to call it Yalawak. And Yalawak is the name for this fish in that region. So we didn't get the last two specimens. There are lots of them out there. And it's actually sister to these other weird things that were just described within the last years as well. Erichthys, which is this guy here, and Cryptodon-Sistris, which is this guy there. If you put that onto the map of South America, we got our Corymbophanes in red, Yalawak in turquoise. Cryptodon-Sistris is actually on the Amazon side of the Ghana Shield, and then this guy way down here. This is not the range of these organisms. This is just where people have been able to get to. There's a vast amount of area here that no scientist has ever been to. When we were up in Kaibaropai, the Patamona there told us, you are the first scientists that have ever been to this area. So that's the kind of places that we try to get to. What is your reaction when you find a new fish species? That's a great question. The writer of the National Geographic article said, if you're so excited when you found this described species, what happens when you find an undescribed species? And so the next day, we discovered Cornboffinese kaii. And as we did it, I'm like, hey, I think this is new. One other guy's like, no, it's just like an ecomorph. I'm like, it's not an ecomorph. And then we came back and showed that it really truly was a different species. So a lot of times we don't even know that they're new species when we find them. So why can't scientists get to these parts? It's really expensive. This Irang River trip cost us about $30,000. It took 4 airplanes to get all our gear into that area. It took 1 airplane to get us out, but as we were flying out, the airplane was giving all kinds of alarms, and we barely got up into the air, and the pilot was like, and to get a bush pilot like that to show a little stress, that's a lot. So, yeah, I mean, this is where we're talking about. There are no roads here. There's no way to get here other than by boat, by airplane, or by helicopter. And we've been all of these weird, really difficult to reach places. All right, the South American darter is another good group. All right, okay. Loading the next set of slides here. These guys are usually about 2-3 inches long, maybe. This guy was about 6 inches, and we were really confused, and we decided to take a look at him. And this is something that's not really that hard to do. We actually have a lot of students do this for us. We actually take pictures of them and then put digital landmarks on them to study how the shape of these things are different from one another. And through our journeys, we got a lot of these throughout this particular region, and we thought, maybe we can figure them out. We call it the Characidium declivirostri group. And so with that morphology, we can see that there are some differences between the species. And that gives us some information that we have different species. Somebody asked if we tag the fishes. We tag the fishes that we take genetic data off of and put into our jars. But as for in the wild, that we can't get back to these areas again, so we don't do that. We've done some of that work in caves and some areas around here where we go back and see if we can find them again. Anyway, here's our preliminary tree of relationships, and actually this crandall is sister to that amila that stopped the hydroelectric plant on the Currabong River. The amila doesn't have these really big pectoral fins that the rest of this declivirostri group has, though, which just shows you that morphology can evolve very, very quickly. Anyway, distributions. Crandall here. One specimen, one dot, one collection from the upper Benchuari River here, because only a few people have ever been there. And this again shows our Proto-Berbice river system. The Declivirostri has a distribution throughout the upper Orinoco River system. This is a new species that we discovered, and there's only like 1 or 2 specimens from each of these dots here, so it's not incredibly common. This thing is really common in the Irang River system up in the upper Irang, but again, nobody had been there before, and so we found it new. So how long are you away from home when you take one of these trips? That original 1998 trip, that was about a month and a half. Most of the recent trips have been a couple weeks. We go in for about 10 days, and then have a few days on either end to do arrangements. Last group are these pencil catfishes, and this is the distribution that we have of our collections of these various species. And this was recently a subject of a phylogeny, and this is where we find one of the specimens that we got. So we actually went in, got specimens from ourselves as well as colleagues, and this is the distribution of all those dots. Somebody asked if the South American darters are related to the North American darters, and no, They were named South American darters because they look like North American darters. It's a type of convergence in morphology, and we see that particular morphology all throughout the world. Are native invasive species a threat to this area? No, nobody didn't put any invasives in this area. You do find down like in the main city in Guyana, Georgetown, you find tilapia there, and you find other introduced species. But once you get into the interior, you usually don't see them. So what we did was several different genes. So we sequenced those and used those genes to figure out how things are related to one another. We put them all together, and all the ones from Guyana here in the Pakuranga Mountains form a group together. And we use this now to actually explore the biodiversity and figure out what species are actually out there. And so we discovered several new species. This elongates. Let's click down here. We found this elongate species up way up here. This is about as far away from humanity as you can get. This is a described species called guianensis. I am going to describe that. But in the Areng River is a similar form, but it's even shorter and squatter with bigger spots. This is really common, but it's found up in the upper Mazaruni here, as well as up in the upper Pataro and Currabong river systems. It's present throughout all this particular area. And it's weird. They can have little spots or they can have big spots. Basically, the big spots are sitting on top of the little spots. So they can turn the big spots off and they have little spots instead. And then there's a plain species that's only up in the upper Mazaruni. That's this guy here. And it's these guys are both related to this widespread species Conradii that is actually related to even to the thing that was way over in Suriname. How long I've been studying fishes? Since 1991. So you can do the math on that. All right, I'm at the end here. The biogeographic history of this scatter shield is even more complex than we imagined. It's taken 20 years to really start figuring out what's going on here. This complex interplay of dispersal and isolation events. And it's important because this area is under threat from gold mining to know what's out there. You can't just blindly preserve things. You have to know what's actually out there. For us, probably Brazil, the Loricariidae River is our next step to get to, but nobody's actually collected in those regions we need to get to. There's a lot of work that needs to be done in Venezuela, but Venezuela, if you've seen the news, is falling apart. So there's a lot of people to thank that have helped us, and you kind of get a little goofy when you get out in the field for too long. Anyway, here's the falls of the Surama, and I will start this video, and you can feel free to text me any questions that you want over the next 7 minutes or so. Oh, technique sampling. That's what we do. This is you set the net, you kick the rocks, and maybe some fish will come out. This particular area does not have a lot of fishes in it. Not diversity, not numbers, and it's really hard to get them out. So I'm sorry this is a little choppy here, but it's a really fantastic video if you see the full res. What was your most successful expedition? That's actually hard to actually put. This particular trip to the Surama was my favorite expedition I've ever been on. It was just so beautiful. Nobody had ever been there before. It's been sort of my goal to get to places where nobody's ever been before, and this was it. There were some humans down there, of course, in Mona, but it was awesome. Do you ever feel in danger? Yes. So that Currabong River, you'd actually have to leap over these channels in the water, and it was really scary because you get washed down, you're gone. What is your favorite type of fish? Plecostomus, of course. That's what I've spent most of my time on. It's a really big group, about 1,000 of them. What's the rarest species you've found? We've actually got a couple. So that one I talked about from the Areng is really rare. But there's another one we discovered based on a collection that would have been made 20 years ago. And that area is destroyed now. So that is probably extinct. Have you ever been bitten by a fish? You can't see it, but I do have a piranha wound on my finger here. Piranhas are really hard to hold on to, and this one got loose a little bit and took a little chomp out of me. And you don't have to worry about it in the water, but when you're holding them, they're really hard to hold on to, and they will get you. The worst though was I got stabbed by a catfish, and normally it's, you make it bleed a little bit, the poison comes out. But this time it hurt so bad. I was just sitting in a stream, just kind of rocking with my thumb like this. It was like one of the worst pains I've ever had. You kind of see the rainforest here. This is right on the border of Brazil, so left is mostly Brazil, right is mostly Guyana. And the stream actually splits. I'm pointing it with my finger. This stream actually splits right up here and has two arms, each with a waterfall coming off of this plateau. I keep sending questions. This is a Chipui out here, and we had an ornithologist around with us looking at birds, and it's just high enough to get the really weird high birds. Do I go fishing in your free time? No, I hate fishing. Fishing is so slow, boring. I like to go out with a net instead. That's what I do for my free time. I like the little fishes. I don't like the big ones. This is the orang itself, and you can see the waterfall right there. Actually, one of the things I like to do in my free time is go out looking for fossils, taking the kids out looking for fossils. What's the largest fish you ever caught for research or fun? The largest fish I ever caught was actually a 5 foot 2 inch long electric eel. That one I didn't manage to get shocked by, but I've been shocked by smaller ones before. And that one, we saw it. We put the net down, and it swam into our net. All the other fish in the net were just shocked to death, but somehow I didn't get shocked. Do you eat fish? Yes, I eat fish. I used to not eat fish, and I used to explain. It's like, oh, you just study fish and you don't eat fish. I'm like, well, anthropologists study people and they don't eat people. But yeah, yeah, I eat fish. Some of the ones down here are really, really good. What's the voltage of an electric eel? They get up to 500-600 volts, but it's the amps that kill you. They're relatively low amps, and people wouldn't die from an electric eel most likely. I've actually touched 3 of the 4 radiations of electric fishes because you got to have hobbies. And by far, the worst one was the electric eel. Electric catfish is not too bad, but still pretty strong. Electric ray, yeah, that one's just a little buzz. How many different places do you visit? I've been in freshwaters all around the world. A lot of work in Venezuela, Guyana, Peru, a little bit in Colombia, Suriname. Been to Brazil, Panama, Central Costa Rica, a few places in Africa, a few places in Asia, Papua New Guinea, lots of places. Are you planning an expedition soon? I wish. I need more money to go back again. You ever feel like you're going to be kidnapped? No, I've never felt that. In some of the most dangerous places I've been, I've never felt safer. There's a relationship that people have with fish and fishermen, and that this is a international language. You can really um, people understand what you're doing. One of the things in that trip, National Geographic trip, um, they asked the locals of what we were doing, and then he's like, they be fishing, and that's what he said. Are there any places you haven't been to that you want to? Yes, in Guyana, there's the New River right along the Suriname border that I would love to get to sometime. All right, I think that's all the time I've got for you. Thank you for indulging me. I love talking about this stuff. So I hope you enjoyed it. All right, I will keep it in mind for next year too.